this first portion is going to be me talking about marine harmful algae identification, and then depending on the time, we'll either break for lunch or we'll just do like a quick 10-15-minute coffee break, and then Amanda will do her cyanobacteria presentation. So we'll just see what time it is when I finish and we'll go from there. So before we dive right into the specifics of cell identification, I'm just going to review with you all, the basics of what is algae, what is phytoplankton, kind of go over the basics of a microscope. I know that Amanda and Sarah are familiar with this. Maybe you guys are as well. I don't know everybody's kind of knowledge level here. So I kind of just wanted to start from the very beginning and go over the very basics of everything. So what is algae? Well, they are aquatic plant-like organisms, but they lack the true stems, roots, and leaves that typical plants have. It's a very large and diverse group of photosynthetic organisms, both multicellular and unicellular, so multiple cells or single cells. They're often referred to as primitive plants, and they're only considered plant-like because, like I said, they're lacking those stems, roots, and leaves that like you'd see a typical plant having. But they also lack those kind of like internal vascular structures that plants use to transport nutrients in water. So if you remember back in middle school or high school when they taught us about like the xylem and the phloem and all those parts of the plant that carry all the nutrients, are algae, macroalgae and microalgae lack those characteristics. They do contain chlorophyll though, like plants. And like I said, there's macroalgae and microalgae, and these are kind of the most common kinds of phytoplankton up here, which I'll explain a little bit further. But phytoplankton itself, I just took the definition, what the actual words mean, derived from the Greek words meaning phyto, plant, and plankton, which means to wander or drift, which is in simplest terms exactly what phytoplankton are. They're little microscopic drifting plants in the ocean. That's the simplest way to put it. There's a lot more to them, obviously, which we'll get into, but that's ultimately what they are. They're photosynthetic. In any environment, they'll float near the top of the surface of the water where the sunlight is. Typically, you can't see them with the naked eye, but they can grow in large enough colonies to be seen by the human eye, which is essentially what a bloom is. When you see, those massive, mats of color that you'll see in Amanda's, especially in her presentation, is essentially what a bloom is. Marine phytoplankton are mainly comprised of microalgae known as dinoflagellates and diatoms, which you can, and then diflagellate. It also consists of photosynthesizing bacteria, which Amanda will discuss, cyanobacteria, as well as green algae, and these armored, I'm not even going to try and pronounce that, coccolithores. So those are the basic, what is algae, what is phytoplankton. Moving on to what are marine harmful algae. Again, this diagram kind of, I think, says it all. They're marine organisms that can produce a toxin that, when consumed by shellfish and or humans and other animals, such as other marine animals or birds, can make them sick or even kill them potentially. And a harmful algae bloom, which you'll hear me refer to HABs a lot throughout the presentation. I'm either referring to, an actual bloom, or we refer to our, harmful taxa that we deal with, harmful species of phytoplankton, as HABs as well. Like, they're harmful algae bloom species. So we'll refer to them. If you hear me say HABs a lot, I'm referring to our specific harmful phytoplankton that we deal with here. So typically, the shellfish are especially susceptible to these toxins because they're filter feeders. So they ingest food by taking it up through the water surrounding them and then filter out what they don't want to ingest. So they'll take in these toxic cells, and then the toxin will remain behind and accumulate in the tissue. And then that can either then accumulate further in other marine animals or it can direct transfer to humans here, as you can see. So that's why it's something that we focus on here at NHDES Shellfish Program, because we deal with shellfish and biotoxins in that way. So phytoplankton is kind of our first line of defense against, seeing if there's potential toxin in the water that could be affecting shellfish and then thus humans. And like I said, these generally, I'll get into this in the next slide, but generally these biotoxins make people sick. I don't know what the studies are and statistics are on, deaths occurring from toxins, but they can, in serious cases, result in death. There are different species of phytoplankton are responsible for releasing different types of toxins that then produce different human illnesses. I know this slide kind of has a lot of information on it. I'll break it down for you. We can't know how much toxin is being released by a bloom just by knowing the number of cells. That's kind of a general statement and saying, you see so-and-so, many cells in the water, you don't know how much toxin is actually being produced. So that's why I say phytoplankton monitoring is kind of our first line of defense and saying, okay, there's a problem here, we need to look further. Which is why you see here we have, this list of target HABs, which like I said, those are kind of our, algae species that we focus on, this list right here. Each has an alert threshold, which is essentially when our routine water sampling of phytoplankton indicates that there's potential for toxicity, that's where that comes from, the alert threshold. So if we're seeing, for instance, if we're seeing Pseudonychia at 15,000 small cells per liter, then we say, okay, there's the potential for toxin in the water. We're going to do some either supplemental phytoplankton sampling, or we're going to initiate shellfish toxicity sampling, which is what we would use to then initiate a closure of a growing area. If that makes sense to everybody. So like I said, we look at the phytoplankton, we say, okay, nothing going on here, we're good. You say, wow, there's a lot of cells in the water, we could have toxin. Let's grab some mussels or oysters, send those to the lab for some tests, which I'm not going to get into all the toxin testing we do because that's a little bit complicated. We can talk about it later if you guys are interested in it. But ultimately, they do these toxin tests, and if those tests reach a certain threshold, then we say, okay, we need to close this area because people could get sick. So that's what these alert thresholds come from. Each of these target HABs, like I said, produces certain toxins. These are the toxins associated with each species here. And then there's a human illness associated, with each of those toxins that's produced. The three main HABs I'm going to be discussing in my presentation, I'm going to go over all of these in this list, but the three that we're going to focus on are the Pseudonychia, Alexandriaum, and the Dinophysis species. These are the species we see in the most abundance. It's where our primary concern lies. And those cause amnesic shellfish poisoning, which is, these all kind of speak for themselves, I guess, and what their illnesses are. We can go over what all the symptoms are of these illnesses later if you guys are interested in, but amnesic shellfish poisoning can cause short-term memory loss, and it can be permanent. Paralytic shellfish poisoning can result in like respiratory failure, which can result in death, and diuretic shellfish poisoning, well, that is obviously gastrointestinal issues associated with that. Something else that I do want to mention here is that you guys probably have heard the term red tide before, and you might hear it in reference to what's going on in New England waters. And I just want to make it clear to everybody that red tide here is very different from red tide somewhere else. Species that cause blooms differ from region to region. For instance, in Florida, you may or may not have heard of red tide down there, where it's been a huge issue for them, where they've lost trillions of dollars and tourism, and they've had a lot of like marine animal deaths down there, and that's caused by the species here, Carina brevis, which we monitor for here as a requirement to look out for it, but we actually don't see it here in the Gulf of Maine, or we haven't seen it here in the Gulf of Maine yet, knock on wood. oh my goodness. But yeah, this is a species that we don't see here, but this is a neurotoxic species that is very common in Florida and has caused massive blooms down there. Here, when people refer to red tide, it's typically in reference to the species called Alexandrium that we'll go over. And it typically doesn't even really, it's rare that you even see color from these blooms. You can have a bloom without seeing any color of discoloration of the water. So just take red tide. I don't want to say take it lightly, but don't automatically assume that it's, oh my goodness, like we have to shut everything down, we can't be in the water. I just think that there's an association, kind of this really negative association with red tide, and thinking that it's something that it's not quite what it is here in New England. So just kind of wanted to make that clear. And then there are two other species here at the bottom, Corinium motoi and Coclodinium polycricoides. Say that 5 times fast. Those we also monitor for. There's the toxic effects to humans are still being studied and not quite known for those two species, but they do have significantly harmful effects on ecosystems. So they can really cause a lot of deoxygenation of water, resulting in fish kills and things like that. So we monitor for them in terms of them being an ecosystem, having harmful ecosystem effects. But they don't, the studies for having negative human effects they're still researching that. So that's just a little bit about the species that we look at. Like I said, the main ones we're going to talk about are Pseudonetia, Alexandrium, and Dinophysis. Next is the stations that we look at. So I won't talk about too much, but I just want to give you guys an idea of, where we collect these samples, how we collect them, just to give you a little bit of background on what it is you're actually looking at and where it's coming from. So you may or may not know that Shellfish Program monitors all the tidal waters in New Hampshire. This map essentially shows all of our monitoring areas. We have two of the stations that we sample for phytoplankton are located offshore. One of them is this kind of mid-ocean site, ACB 30, that's between kind of the Piscataqua River Inlet here and Isle of Shoals. And the second site is the Isle of Shoals area in Gosport Harbor. We also sample UNHPR Coast Guard Station, which again is kind of right here at the inlet of the Piscataqua River. We also sample in Hampton Seabrook Harbor. There's a dock right in here in this harbor that we sample at. And another site that we've picked up pretty recently, sampling regularly, is in Lower Little Bay. It's right adjacent to a commercial oyster farm. So all of these sites were chosen based on not only their accessibility, but also their proximity to active harvesting areas, whether they're commercial or recreational. We try and do these year-round every week. Offshore, that I gotta say right up front, that does not happen. We get out there as much as we can, but the sea conditions, especially in the winter, definitely don't allow that. So, but the sites that we can sample by land, HHHR2 and UNHPR, we do sample every week throughout the year. Let's see, field collection. I realize I'm in most of these pictures, not by choice, just these were the best pictures we had to show what we do in the field. Basic collection, and I can show you, we have all the sampling supplies here, and I can show you guys like a walkthrough of how we sample if you want to, but this is something that I don't think you'll ever need to do, but like I said, it's just good for you to have kind of some background information on like what you're looking at under the microscope and where it's coming from. So collection ultimately happens, we have this weighted bucket that Chris actually kind of just made. He just kind of attached Chris does it a lot. He makes some of our equipment. It's good. No, it's good though. It's literally just some of these, what kind of weights are they that you use? Oh, yeah, okay, so he found some weights and like attached them with zip ties to a bucket so that it sinks when we put it in the water. And so we lower this bucket about 1 meter below the water surface and then pull it up and collect 10 liters of seawater. It's kind of supposed to be like a composite sample, but it's not really. It's a very efficient method for us to use because we have multiple sites we do at in multiple locations, and it's just, this is kind of the method that we've adopted, and it seems to work for us pretty well. It's pretty simple and straightforward, so that's what we do. So like I said, we pull up these 10 liters of seawater and we pour them into carboys. We do it twice. We have one like original sample, and then we always have a duplicate sample just in case something happens to that original sample. You spill it at the end or something. It's happened before, so. And then filtration happens where we take those 10 liters of seawater and pour them into this PVC device, which is this white. You can see Eric's holding one here, and then I'm holding one here, and there's one right here as well. These kind of white, again, like makeshift PVC devices where on the bottom of it is a 10 micron sieve. So it's like a very small, fine mesh, essentially, that collects, as you pour in the seawater in, most of the water will filter out, and then it'll collect the small particles or phytoplankton that will then stay on that mesh. And so as the filtered water is coming through the mesh, I'll collect that into a spray bottle and use that spray bottle to backwash the sieve to obtain 15 mils of a concentrate. So I'll let all the seawater filter through, and then what will be left on that 10 micron mesh or that sieve is going to be just kind of these particles. And I want to get, at least 15 mils of water with those. So I will just backwash the sieve and with that filtered water and collect 15 into the sample vial like I'm doing here. And then probably the most important part, one of the most important parts, is that we need to preserve it with Lugols. The other preservatives you can use, Lugols is the easiest for us to use. It's not toxic, and it's not really that toxic. And it does stain, though, I will say that. So if you ever use it, it's iodine. I'm sure you guys may be familiar with it. But we want to use about 4 to 6 drops of Lugols for preservation, because otherwise all the copepods or the zooplankton are going to feed on our phytoplankton, and that's what we're trying to look at. So if we don't preserve it, by the time I look at the sample, there might not be anything in there for me to look at. And then we store them in a cool, dark environment because the Lugols are light sensitive, so you want to make sure that they're kept in the dark. And typically the temperature where you store the samples isn't as important as the dark, but you don't want to keep them in like a really hot environment. You kind of want to keep them at ambient temperature. So that's kind of the collection. And Chris is bringing in some of the sampling supplies if everybody wants to see. All right, moving on to Microscope Basics. So I found this little image online that just kind of goes over the basics of a microscope, and it looks very similar to the one I have here, which you guys are free to look at during break or whatever. And you guys, I'm sure you've all seen a microscope up close, but just to review, you have your eyepiece lens, which in this picture there's just one, but typically there's two. I don't think I've ever used one with just one eyepiece lens. I think Amanda's has two as well. So there's two eyepiece lenses here. You've got this large black rectangle here, which is the stage, and that is what holds your slide, which contains your sample. And you can move the stage up and down and back and forth so that you can view all the parts of your slide, which again, I can show you all in my microscope what that means. You also have this lamp here, which illuminates the slide so that you can see it. And you have a focus wheel on the side, which the smaller knob is fine focus, and then the larger knob is coarse focus, which typically you don't really need to adjust the coarse focus. It's coarse focus. So if you move it even a little bit, it really changes the view of the slide. But the fine focus you'll use quite often to, kind of see different parts of a cell, which again, it's hard to explain until you actually see it, but I can show you examples of this later on. So those are kind of the basic parts of a microscope, if that is helpful to anybody. Okay. So the counting chamber that we use, which I think is also what Amanda uses, so this is it's good that we use similar things, is called a Cedric rafter, which is kind of known as like a low-tech method for counting phytoplankton mixed assemblages. It's easier for counting and calculation because there is a known volume in this slide. It fits exactly 1 milliliter of a sample. And there's, as you can see, there's grids on it, so you can count a certain number of grids or columns or rows or however you want to format it. You count a certain number of these grids, and then you can calculate your cell density based on that. So it's a pretty easy way of determining cell density. So that's what we use. To load it, you do exactly as this picture is telling you to do. It's important to invert the sample vial a few times before you actually load the Cedric rafter, because you want to make sure that you're homogenizing the sample. By the time you pull, you very gently invert it a few times. You don't want to shake it at all. You just want to have a nice little demonstration here. Invert it a few times. You then can use any kind of pipette that you have. I just have, plastic disposable transfer pipettes to pull 1 mil from the middle column. So like I said, you want to make sure you're not pulling from the very top of the sample at the very bottom, just kind of right in the middle. You pull 1 mil from there, and then you slowly introduce that into the corner of the Cedric rafter. The cover slip sometimes it will slowly move as you fill the sample up. Sometimes you have to nudge it with your finger a little bit. And you just want to do it slowly and try not to overfill it, because like I said, it holds an exact volume. So if you overfill it, then the cover slip won't suction to the sides of the Cedric rafter. It'll just kind of like float on top. It's actually, I feel weird saying this, but it's actually more difficult than it may seem. Sometimes there's air bubbles that get stuck, and you want to try and avoid that, but it's not the end of the world if you get an air bubble here or there. So like I said, you just want to fill it up, which again, I can walk you through how to do that in a demonstration. And then you want to let it settle for about 10 to 15 minutes, because after you've homogenized it, you want to let it settle again, because now everything's kind of free floating in that slide. So before you start counting, if you start counting right away, you're going to see things drifting. So you kind of just want to wait till everything settles. And the first thing you want to do is you can use the lowest magnification, which is 4x, and you want to just do an initial scan of the slide and say, okay, can I actually count the cells in this slide, or do I need to do a dilution? Or perhaps I need to look at a different type of sample. Like perhaps I need to look at a 2-liter concentrated to 15 milliliter sample as opposed to the 10-liter concentrated to 15 mil sample. I'm thinking typically if I give you guys samples to look at, it'll already be diluted or have been in a format where you guys can actually count. You don't need to do these kinds of extra steps. But it is good practice to just look at it and say, okay, can I actually count this? Because there will be instances where it's so full of either a Hab species, or it could be a non-Hab forming species, which I can show you an example of, or it could be just sediment and organic matter that is it's obscuring your view so much that you need to dilute, which has happened to me a couple times. So that's something you can do before you start. And then this walks you through specifically how we count every single time our cells. It's very specific. I'll walk you guys through it. If you're confused, that's okay. I have this all in an SOP that I can give you. We can walk through an example together, all of that. But essentially, you want to adjust the stage to the bottom left, which is in microscope view of the Cedric rafter. I say microscope view because it's opposite, it's inverted. So if you're looking through the microscope at the bottom left, it's actually the top right if you're looking at it, just looking at the stage from outside the microscope, if that makes sense to anybody. So you're doing it bottom left microscope view. You count 5 columns to the right, like here, and then you start counting on the first column. This is considered column 1, which is technically the 6th column over. And that's column 1 for the analysis. You then skip 2 columns, and then column 2, skip 2 columns, then column 3, and you do this until you've reached 10 columns. So in total, you've counted 200 grids, or 0.2 milliliters, which you can then use to calculate your cell density, because like I said, you have a known volume that you counted. You counted 0.2 milliliters of a 1 mil sample. So from that, you can determine your cells per liter, which I can show you a quick reference table I have later on for how you calculate these cell densities. I do my counting at the 100 magnification, which is the kind of medium power. And you can shift to 400 to confirm identifications, which is the highest power you can use. My microscope has 4 objectives. You cannot use the 100, the 1000 power, because it's too long that it'll break. Like if you try and use it, you end up breaking the Cedric rafter, because the Cedric rafter is so thick that if you try and move that objective lens over, it'll crack the cover slip. So you just want to not use that one at all. It might even be easier if you're not used to. I've done it before. I've broken, just when I first started, I would swing them around and I accidentally cracked one. And if it's easier, you can even just remove the objective to keep yourself from having that issue. And I just say take pictures and use the guides, because that's what you're going to need, at least when you first start. And even when you get good at it or become more experienced, I use guides all the time. I Google all the time. It's just, if you're unsure, which happens frequently, you just want to use all of the resources that you have, which I'll provide you with a good list of those. And yeah, like if you can take pictures, I mean, this microscope here I have equipped with recently equipped with a camera that I can use, that can take pictures and that I can load onto my laptop and then share with people. For you guys, if you guys show interest in this and we figure out microscope that you're going to use, I think Amanda has a couple, she said, in the lab that you guys could use. And I believe, Amanda, you have camera attachments for those? Okay. Yeah, and there's a possibility that you guys could be using this at some point, because we're supposed to be getting a new microscope as well. So we can work out the logistics of that a little bit later, but we have some options for you guys if you ever want to practice. And I think the taking pictures is highly useful. I actually just did it recently. There was a cell where I was looking at in the microscope and I said, this looks like it could be like a harmful species, but I really don't know, and I don't feel confident identifying it as such. So I took a few images, I sent it to this whole list of, colleagues that I have that are all pretty much HAB experts. And I got a bunch of replies saying, I think it's this, I think it's this, I don't think it's what you think it is. And it really, I'm just so thankful that I had that community to reach out to, because it really confirmed my thoughts of, this isn't the harmful species I think it is, but it could be, this or that. So it's just really good to have those images to share. So that's general counting guidelines. Quick review of, this is my little makeshift slide of, what do you count as in or out? Because like I said, the Cedric rafter has these grid lines. So you want to, if you're counting a column and you see a cell and you're like, wait, is that in the column I'm counting, or is that not in the column I'm counting? This is kind of a little guide to help you figure it out. It's pretty straightforward. Most of all, you want to be consistent. If you're doing it one way, do it that way the entire time you're counting, and for every sample that you're counting. You want to be consistent. The way that I do this is that if it's in the column like this, of course, so if we're counting this gray column here, if it's in the column like this orange cell, of course we're going to count it. If it's partially in like this orange cell, of course we're going to count it. If you have a chain of cells, which is common with Sudnichia, as you'll see, you can count the ones that are in the column, but if it's outside, like this blue one, you wouldn't count it. If this blue one's partially in, then you could count it. These cells here, like this blue one and then this blue one, I mean, if it was moved over a little bit more and it was a percentage of it was in the cell, I would probably count it. some of this is going to be analyst discretion, and you know, you all are new to this, or a lot of you are new to this kind of counting method and just cell identification in general, so it might take you a few tries to say, okay, I can't really tell if that's in or out. Ultimately, I would say if you're really not sure about one cell, just go with a more conservative approach and say, okay, that's probably in the column. That's usually how I would do things if I really don't know. But typically you'll look at a cell, there'll be very rare occasions. I shouldn't say it won't happen because it will, but there will be rare occasions where you'll look at a cell and say, I don't know. Like it's usually either in or out. So like this orange one here, I'd say most of it's in. I don't know if this is somewhat helpful to everybody. Yeah.
Expand on what you said. The reason that Brooke is encouraging people to be conservative, and if you're not sure, just count it. What we kind of try to remind everybody that is doing this is that when you're counting phytoplankton, you're not really making public health decisions. You're not saying this area is going to be open. This is really to trigger us to know whether or not we need to go test the shellfish for biotoxins. So that's really so. If Brooke comes back and says, you know, I'm not sure, but I add a lot of extra cells, the worst that happens is it pushes us shellfish, and we spend $200 or whatever, and then we find out there's no toxin, and we're good. And so that's the reason for doing that. And the other thing I wanted to mention is that while Tiffany was here last year, Brooke, Eric, and Tiffany did a lot of work where they would take one sample. Like Brooke would go count, she'd take that centrifuge tube, she'd take the one mil out, and she'd do her counting. Then Tiffany would take that same tube but not count that same slide. She would get a different 1 mil, and she would count. And of course, they never get the same number, right? And we were doing that on purpose. One, early on, it was to help Brooke develop confidence, or she's like a newly trained person but has not done this a lot. And by comparing her results to Eric, who was very experienced, she started developing, oh yeah, I know what I'm doing, and I'm really good. But it also gave us a bunch of comparisons so that we would start to get a feel for, you know, if Brooke says there's 700 cells per liter, how tight or fuzzy is that number? And now we have some quantitative measurements of how fuzzy these numbers are. So when you're counting, one of the things that we're going to talk about is if you choose to count the stuff we leave for you, Brooke has already counted it. You'll have access to her results. You'll get your own results, and then you can see how close or how far you are. And by using those comparisons, we can tell you if you're within, 100 cells, you're good. I think early on, both Brooke and Eric were freaking out because, oh my God, I got 1,000. Right. I only got 800. We're like, we don't even know if that's good, bad. Yeah. What does that mean? So as we get into this, if we want, we can sort of share that.
Yeah, I think, and that's something that we kind of faced this past year was, can you do quality assurance or quality control with phytoplankton enumeration? And it's really difficult, and it's something that I don't think a lot of people do because it's so hard to control for all of the factors that go into it. So I guess essentially, and what I say in the SOP is that we try to control for as many factors as possible so that when we do some kind of quality assurance measures, there's consistency and there's some standards to go against. And this is one of them, to just make sure that if we're all counting the same way, even if it's a small, cell here and there, we're all being consistent, and that's what we want. And that's also why the slide before I say, okay, we're going to count 5 columns over, count this column. At least we can say, okay, we're all counting these grids the same way. We're counting the same number of columns. There's some consistency and some standard measurements to go against. So that's kind of why it may seem so stringent, and that's why we're just trying to control for as many things as possible. So this is just another example of one of those things. And then this is the quick reference table I was referring to. It may look kind of complicated, but it's actually, if I show you the math to how we got to these calculations, you'd pull your hair out. So this is a really easy, it's I don't know. But, and it's if you want to see the math, it's in the SOP. All of the, methods behind all of this is in the SOP. But essentially, if you're counting, note this up here, that if you're counting 200 grids of a Cedric rafter slide, which you typically would do, of a 10 liter to 15 ml concentrate, which is what you typically would be looking at, all you're going to do is take your number of cells, multiply it by 7.5, and that's how you get your cells per liter. So if you see one cell, it's 7.5 cells per liter. Done. That changes as you dilute or use a different kind of filtration method, or if you're using a whole seawater. Again, math, you can see that in the SOP if you really want to see it, but essentially all you have to do is multiply by these numbers that I provided here for you. But like I said, this circled one up here is what you're going to use 99% of the time. And now we can get into the cell identification, which is why you're all here in the first place. But I just had to walk you through all my fun little spiels. So just for fun, these are some live images. Most of the images I'm going to show you, or a lot of the images I'm going to show you, are still images that, like I said, Eric captured. And I have a massive folder of just about every species we've ever seen, or he's ever seen, that I'd be happy to make available to you all. I don't know if it's very, he had to give it to me on like an SD card because it was just so packed with images. But it is very useful, and if you're trying to determine, what is this? It's in his list somewhere. So this is just for fun. This is Cattosaurus, that's right here. This is Protoparadinium, which is actually, that's my favorite kind of cell. I don't know why, I just think it looks really cool. So yeah, these are actually images that he took, which I think they came out really great. So first thing, before we get into cell identification, I do just want to mention quickly that cells are going to look very different depending on the angle that you're looking at them, which is something that I don't think I realized or other people may realize when you first start to look at cells, is that you're not going to look at, this flat object, this 2D thing. It's not going to look the same multiplied, It's when you're looking at a diatom, for instance, you can have a valve view or girdle view. And so a valve view is when if I'm like facing the front on the microscope, that's valve view. If I'm facing the side like this and you're viewing my side, that's girdle view. So that just shows you how different, like that, you could look at that and think it's two different species, because why wouldn't you upon initially looking at it, But it's not, it's just different views. Same thing with, potentially a dinoflagellate. You can ignore all the numbers. I know that's a lot, but essentially the picture shows you that when you're looking at the dorsal surface, which is the back, so if I was turned around, that's dorsal, it looks very different than ventral, which is the front. So that's, just a little bit of, how they can look different. And then the same thing with you look at the anterior, which is the top, or the posterior, which is the bottom, which you'll see another example of that. But that looks completely different than ventral and dorsal. So it's just another added layer of difficulty to identification. Especially, some of the cells that we look at, you'll see it doesn't necessarily matter what angle. You can pretty much determine the species, no matter what angle. There are some, particularly Alexandrium, where the angle, it really matters. And Alexandrium is the most difficult one to identify, and you'll see why. So we'll get into Dinophysis. It's a dinoflagellate. It has these are not really technical terms, but it has what we call kind of a crown and then a back fin. So this is the crown here, this right here, and then the fin on the side. I think it looks like a blimp. I often, when I'm identifying things, I find it's very helpful to kind of compare them to everyday objects. So if you hear me do that throughout the presentation, that's just what I find very helpful. And I think it is for other people as well. Because then you say, oh, Dinophysis, that's the blimp. Trust me, it helps. So the species that we see most common are Acuminata, which is a small one here, and Norvegica, which is this kind of pointed one here. We also see Tripos, and we see Forte as well. Those are the most common ones we see. It has a flagella, which is kind of that hair-like whip or tail, but it's not always apparent. I don't usually see it when I'm looking at this species in particular, but it does have a kind of whip. The morphology can vary a lot, as you can see in this picture here, how some of them look very similar, but then some look very, very different. It has a cellulose shell with pores, which it kind of shows you in this image as well. If you at certain angles, certain focus, you can see this kind of these pores here. And then this is just another picture of Acuminata, which it looks pretty similar to this. Acuminata is a lot smaller than the rest of the species, which you can kind of tell from this image as well. And so, okay. So the next species, which, like I said, is the most difficult one, is Alexandrium. And this is another really great diagram. I know that the numbers on the plates can be confusing, but it is an armored dinoflagellate. So it has these thecal plates. And this is just showing you how they have them numbered a certain way, which you don't have to know that. But I did like this image because it shows you, the ventral view here, and then you have dorsal, and then you have, anterior and posterior. So you can see how if you look at it from even the bottom or the top, this doesn't look anything like A and B, which is, to me, how it can be so difficult to identify these. And sometimes what I do after I've completed a count and I have cells that I'm not sure are Alexandrium or not, I'll note which column they were in, which grid they were in, and I might go back and tap them a little bit. Be very careful, because if you will break the cover glass if you tap too hard, but if you grab like a toothpick or something, you just gently nudge the cover slip, you can kind of try and rotate the cell so that it's at an angle like this, where you can actually see, more of the features like A or even B. Because there'll be times where you look at it, I said, in C or D view, and you have just no idea, and you need to adjust it a little bit. So there'll be times where I might go back and just poke at them a little bit. They may or may not have this kind of peak right here. Not all of them have this tiny little tip. They have what's called a cingulum and sulcus, which are essentially just kind of these grooves. It's like a girdle. They call this a girdle too, as you can imagine, kind of just wraps around the middle of the cell. And then the sulcus is this part right here, which is like a deep groove. So you can even see in this image right here, like you can see where the cingulum would be, right where right where it indents here. And there's kind of a little peak, but not too pronounced. Then you can see where the cingulum would be right here as well. So that's actually it's a really great picture of an Alexandrium cell. They're not all like that. So again, I'll take you through some images and we can just talk about what we think they are.
So there are a lot of different marine species that look like Alexandrium?
Yes. Yep, and I'll show you, there is a guide, actually, I don't know if I can I can click it here. So this guide, I'll see if this works. And I have this in, I was looking at pizza. I have this at the end of the presentation as well, a link to it. But this is a guide from Maine DMR that I think is very helpful, and it tells you Alexandrium is very difficult to identify, and it's helpful to know what is not Alexandrium, because you will see a lot of what's called Scripsiella, which is very similar. In a lot of ways, it doesn't look at all like Alexandrium, but at certain angles and some cells look very, very similar to Alexandrium and can be confused. But this document I think is helpful. And like I said, there'll be a bunch of other guides that I'll provide you with that will show you cells that can be pretty similar looking. But I think Scripsiella, in my opinion, which is the cell, is the one that looks the most like Alexandrium, is most often confused. But you see Scripsiella in abundance. Like I see this in almost every sample I look at, and sometimes in huge quantities. So that's important. If you're gonna get them confused, like that's just important probably not to, because it's very common. So yeah, that's a really helpful guide. That's from Maine DMR. So yeah, we'll go through.
Like that last species you just presented had like a clear sheath around it. Does Alexandrium ever have like that clear sheath? at least in my guide, it looked like there was like a clear kind of opaque.
Oh, like in the Scripsiella thing?
Yeah.
I don't think so. Well, I don't know, because I've seen some images, I've seen some cells that kind of look similar to that, and that we have thought were Alexandrium. But typically, I don't know, my answer to you is I don't, it could be. It has happened where it's kind of it like expanded almost a little bit. And it's just like the cell itself is still like smaller within that. But and we've actually had, I think there was last year, we saw a few that looked very similar to the Scripsiella, but didn't have that point, and we were like, this could be Alexandrium, we're not really sure. But it did have that kind of sheath, and that wasn't something that I normally saw with Alexandrium, so I wasn't sure, and that was, so we kind of just marked it as uncertainty, which is something else that I do with Alexandrium is, because they're so hard to identify, I've kind of implemented this. If you have greater than 50% confidence that's Alexandrium, then mark it as such. If you're like, I really don't know, but I don't think so, and you have less than 50% confidence, then don't mark it as such and just make a note of it.
Sort of backup for all of this Alexandrium is that this is the one harmful algal species where no matter what Brooke and Eric or whoever see, we're always testing the shellfish for this. Because this is a very common problem for us in the spring. And because they were having these problems, and it's very common across the Gulf of Maine, so it wasn't limited to Brooke and Eric.
Yeah.
I'm not sure. You know, like Beatty and Mars got a ton of experience doing this, and they have the exact same kind of testing.
So when you're testing, though, you're not testing for the presence of this particular species. You're testing for the toxin.
Yeah.
So what is going on? What used to happen in New Hampshire before we did phytoplankton monitoring, because our life was really easy before all these crazy conditions started changing in the Gulf of Maine, is we had Alexandrium, and almost every like late April, early May, they would bloom, and sometimes they would bloom to the point where we would see toxicity in the shellfish. Lots of different types of saxitoxin. There's like a lot of different flavors of saxitoxin. But if there were enough cells in the water, it would show up in the mussels, and then they would do the closure, and then the bloom would die off, and eventually the mussels would be fine, and we could breed them. And that's all we did. So we would collect 2 blue mussel samples a week, one from two different places, and we would just keep track of it that way, never knowing what was actually in the water. But now that we have to deal with things like pseudomycia, which we never really had very much of, and dinophysis, there's new species of these things popping up that are like super toxic. That's why we have this phytoplankton program. We didn't have this in 2015, it was 2016. Where we asked Eric, who had been doing this as a volunteer just because they thought it was fun, to actually be a part of the NHDES program, because it's becoming more and more problematic across the country. And now we're actually required to have these phytoplankton programs. And Brooke, I think, mentioned that we do this year round because we allow harvesting in these places. If we're allowing commercial harvest, we have to be testing. Even if it's a time of year where we usually don't see anything, we don't have a choice. We have to do the testing. So, but it's really instructive and helpful for them to know what they're looking at, whether it's Alexandrium or not, because we are trying to build a sense of how many cells does it really take before we start seeing toxicity. Brooke said we're being super conservative with this. If they see 1 cell, they report 75 cells per liter, and that's enough to do the test. But we're doing the test. So, like, if you guys get into this, I think hopefully one of the things we're thinking of, we want to talk with you about, if you want to continue with this, is we're going to target, like, when we get to April, and she starts seeing Alexandria. We'll try to be communicating with those of you who want to sort of do this on a monthly basis, and we'll make those particular samples available up in the Linden Center, so you'll have a chance to look at it. Or you'll get a chance to see one that she said 50% confidence. I really don't know. But we'll sort of talk about those.
Yeah, figure it out. Yeah. No, that's helpful. Thank you. Yeah, like Chris said, this is a species that there is a sense of a little bit of relief because we do test for weekly, at least for half of the year. we test weekly at a couple sites. So again, we'll do our little fun activity that I know you guys
So can I
love.
ask one more question? I'm gonna ask a question.
Please do. No such thing.
Because I don't know. So if you, that particular species, and you then go out and test the shellfish itself for a neurotoxin, is the neurotoxin, the presence absence of that neurotoxin specific to that species? So if you see it, you know that species is there, or something else also could have caused it?
So do you want to answer this? You want me to?
Yeah, so especially with Alexandrium, we don't identify it. First of all, we don't identify the species, so it's just that genus in general. But if we do tests where we can identify presence or absence of the toxin in general. And then if there's no toxin present, then we don't go any further. If there is toxin present, then we will go a step further and do an additional testing that will give us a certain number of toxicity and based on requirements that we have in the
So
levels.
The toxins aren't necessarily specific to one genus. Maybe.
It
Yes, that's-
would
So
be,
that question depends on what you're asking about. If we're talking
I
about
think-
ESP saxitoxins, there are but there's not one saxitoxin.
Yeah.
There's a bunch of different ones. And if you were in different parts of the Gulf of Maine, Alexandriaum cantonella are present everywhere, and they all produce saxitoxins, but the congeners that they're producing in closer to Nova Scotia are very different from the ones down by Mass Bay. And this becomes a problem depending on how you're testing for the toxicity. We in New Hampshire are like savages, and our test is the mouse bioassay test. So we give them the mussels, they shuck them, we grind them up, they boil them in acid, they extract it, and then they inject that into mice. And the toxicity.
I was gonna leave that
how long
out.
does it take for a mouse to die? Like in seconds.
Yeah.
And they do that.
Is that what those bags are?
We are doing that, and we're probably moving away from that.
I hope so.
Like, that's just been, now that test does not tell you anything about the content.
Right.
Like,
Is the
if
toxin
mouse is
there
dead within
so many
5
hours?
seconds,
Yeah.
then the table says that is 130 mouse units per 100 grams of tissue. Now, Maine used to do that, but their volume of testing is huge.
Yeah.
And, they've moved away from mouse bioassay, and they're doing HPLC. So in that, you can actually see what the different flavors of saxitoxin are.
Yeah.
We are now moving in that direction for New Hampshire because the new lab staff are just like mortified that they're doing it. And we actually bought an HPLC machine with red tide disaster relief funds 10 years ago. And they kind of were playing around with it and they're like, you know what, this isn't going to work for us because you don't do that many. You have to keep like the standards up and all this other jazz. And we only do like 2 seconds a week. So they're kind of like, yeah. Now they are trying to revive that. But in the interim, we've got these like cool screening tests. They're like home pregnancy kits, and they do just a presence absence. It's there, but it's not going to tell you much. So we're not slaughtering quite as many mice.
We're trying to get more of those. Yeah, those were great. Those screening
But now, if
kits.
you were doing something like, we weren't talking about parallel shellfish poisoning, we were talking about amnesia shellfish poisoning, which is the pseudonychia. There's something like 52 different species that have been described in the Gulf of Maine, and not all of them are toxic. And the really pain in the backside about pseudonychia is that we can have, Brooke's going to show you a slide in there, and there's just like a ton of cells everywhere. Just because it's like 1 million cells per liter doesn't mean that they're producing any toxicity
Right.
at all. Like, they don't tell you when it's going to start, which is the scary part.
Right.
They don't like light up a certain color when they're producing toxin. Yeah.
And then the diaphysis ones, now we're discovering there's not just nucleic acid, which is its toxin, but there's another compound that's screwing around with the tests. It's really, it's like a crazy type thing to get into. So, is that, I don't know if I answered your question.
So, yeah, basically when we get results, it just tells us that the toxin's in the water, and we relate it to, the Alexandrium genus. We don't, like Chris said, Cantonella is the most common species, I think, but we can't, with a light microscope, it's really nearly impossible to identify the species, especially with, you'll see with Pseudonychia, we did not identify the species with that.
Related to that,
Yeah.
do Is it known whether the toxins can just be free in the water, like after one dies?
Oh, I don't know. It's a good question. I can do some research on it.
I'm just curious. That's what happened to cyanobacteria. As it dies,
Like
it can be,
aerosolized
like, a
or
free toxin,
yeah.
like a chemical in water.
I don't know with Alexandrium if that's the case.
The question is Alexandrium, is the toxin aerosolized? Is that?
Or no, it can
Or just
be,
persist in the
like,
water.
can it leak out of the cell and be like a free extracellular toxin in the
So
water?
Yeah, with Alexandrium, not so much here, and the reason for that is that those fecal plates that Brooke was showing, they're pretty tough.
Yeah.
And a very similar species to our Alexandrium exists in the Gulf of Mexico, and their plates are not tough at all. And so when they have these blooms, they have to warn people, you're going to get skin and eye irritation walking on the beach because these cells are in the surf and the surf.
The waves crashing.
And all this stuff is like the reason. We don't so much have that problem.
Yeah.
For that one, I can't really speak to the other ones because we're still learning how to deal with
Yeah.
pseudomycia.
Yeah, if I'm being completely honest, there's a lot I don't know. Like I'm giving you my spiel, but there's so much I still don't know, and I'm still learning about all of these. And they're still doing so much research. Like I said, with those two species, Carinia, Mikimotoi, and Cochladinium, they're still doing research to see if it even has harmful effects on humans, because right now they just know that it affects, finfish and shellfish negatively. So it's just an ever-evolving, always learning field. So, okay, on to the last major tab, Sunichia, which can be relatively easy to identify. This is a pennant diatom, which is like a boat shape almost. These occur almost always in chains. You will see like them in single cells, but they're very commonly in chains, usually in like even numbers, like 6 or 8. They have kind of these pointy ends, or truncated ends, and they overlap in kind of the stair-step fashion, which you can see in this middle image. But I actually like this image a lot because it shows you how a different view, the stair-stepping looks differently. So if you're looking at it like girdle view, it's, you've got the stair-step look, but valve view, you see the overlap, but it doesn't look quite the same. Does that make sense to everybody? This is overlapping, but it doesn't look the same as this overlap here. So those are just different views. Yeah, there's 2 chloroplasts on the side and then kind of a clear center, which is the nucleus. They can be very variable in length and width, and we don't distinguish, like what Chris said, I think there's like over 60 species now of Sunichia. It's impossible to distinguish with a light microscope. You'd have to use like a scanning electron microscope or something, like much higher technology to determine species, but we do try and categorize them based on their width, which is the transapical axis, which is kind of the fattest point of the cell. So right here in the middle is where you'd measure, and I'll show you guys an example of that in a minute too. We consider small cells anything that's less than 3 microns, which I'll show you on a scale, again, what 3 microns is. And a large cell is greater than or equal to 3 microns. And we distinguish these and have different alert thresholds for these different sizes because small cell are likely to produce less toxin and large cell are likely to produce more toxin. So that's kind of just a general rule that we go by. We used to identify small cell, intermediate cell, and large cell, and that just got to be very difficult, and it's just too time consuming and too much of a headache for us to deal with. And so we kind of just lumped the intermediates in with the larges, and yeah, that's how we categorize them. Here's another picture of, these are also, this is great. This picture here. All these little, like what look like scratches are Pseudonychia.
Is that for it?
This is, I think that's 10. Yeah. No, yeah, yeah.
There's all sorts of ways we can make you feel old, like you can't believe.
Oh, great. Yeah, no, that's 10, I think. Yeah, but you can see like this is a like I don't know if you can see this is a large right here. This is a large. This is a large. So there's some larges mixed in there, but all these little what look like literally look like scratches are small cell
This is
synochius.
why if you develop this as a skill, you will be highly sought after.
So that's fun. Here, this is a great example because this shows this is Synoechia next to Rhizoslinia, which can be confused for Synoechia, but it's not. And I think it's once you see them next to each other, it's pretty obvious, but or I think it's obvious, maybe it's not, I don't know. But there's a lot more like chloroplasts here. It's a lot more elongated, and you can see these very like pronounced pointed ends here, which I can show you another image of that, but it's very different looking than Synoechia in a lot of ways. But this is a large cell Synoechia, and you can see here, you know, the chloroplasts on either side, and the little clear circle center, the nucleus. You can see them overlapping, kind of on. This looks like the valve view. Like you don't see that stair step overlap, but they're on top of each other. Measuring cells. This table, I know it may seem complicated. All you really need to know is that if you're measuring cells, you want to do it at the highest power, which is 40x power. It's the blue. That's why it's blue, because it's the blue on my microscope, the blue objective. And what I have in my microscope is called a reticle, and it's basically a scale, like a very, very tiny scale that you put into the eyepiece. So when you look through the microscope like this is what it would look like. You can see this scale line up in your eyepiece. And this allows you to measure cells. And so typically, if I'm unsure of whether something's large or small, I will use the scale. I don't use it for every single identification, because there's a lot of times where you look at a cell, that's definitely large, that's definitely small. But there are times where you can find those kind of intermediate sizes where you're like, I'm not really sure. So I'm gonna measure. And kind of my general rule of thumb, well, each division, each of these very small divisions, and there's 100 of them, is 2.5 microns, or micrometers, same thing. And like I said, the small cell is less than 3 micrometers or microns, and large cells greater than 3 microns or micrometers. So typically, my kind of general rule of thumb is if it's smaller than 1 division, like this one is, or it's about the same size of 1 division, I'll count it as a small cell. And if it's larger than 1 division, like this one is, then I'll count it as a large cell. Some of it is going to be up to your own discretion. It's not, it's as exact science as we could get it, as I can get it to be, knowing, you know, that we have the scale available. And if you guys don't have one on, I don't know if you have one on your microscope, but we can easily order one, make it available for you guys. And you can use this one as an example just to see what it looks like. But this is actually this is the exact view that you would see, is this, you know, these 10 large tick marks and then all the small divisions between.
Amanda, do you guys have something like this up there, or should we?
Oh, we do, and also the Nikon scope has a camera that's calibrated, so you can measure on the like iPad-like thing.
That's even better. But this table just shows, like, you know, under each magnification what the division would be, but you'll always do your measuring under the highest magnification. So what you need to know is that each small division is 2.5 microns. But if you want to know for fun, the other ones, they're here. And this is also in the SOP, which I will have a link to, and I can print out for you guys as well. So everything in this presentation, plus more, if you can believe it, is in the SOP. So don't worry.
That's not enough for you when I finish. Finally, Tiffany brought the clap almost to the finish line. And decided she wanted to work. So I haven't quite got to that, but we just have to put a few more things in. A lot of the supporting info in the SOP is going to be.
Yep.
Really,
Because I know this is so much fun. It's fun for me.
was
But
initially started by Karina back in 2017.
the authors on it, or there's like 5 authors on it, because I'm on it, and then
The
Chris.
program like kept them all evolving.
Yeah,
and
it's
then-
changed
so
so
like, we don't
much.
wanna finish this until we finally.
Well, yeah, the SOP that I rewrote the SOP pretty much completely because the original one Eric had brought in, and a lot of it was accurate in terms of the field collection, but our analysis has changed so much over the years, especially because we've seen blooms past couple of years that we had never seen because we hadn't had a program, and also because we just hadn't had blooms in the past. So we had to adjust a lot. I think it was 2020, we made a lot of changes because we were seeing things that we weren't equipped to really handle. But now we are hopefully equipped to handle them. This is just another example of, it looks so tiny on the, but that's what it looks like. I mean, this is 10x. This is like the medium power, which you would do your counting on. You move to a higher magnification, for your measuring and to get, clarification. But, this is a lot. Like I can, just looking at this, I can tell this is a large cell. This is clearly a small cell. And you got this one over here where you're just, I would probably measure it. Looks like it could be small. I don't know. I've done this I've done this a lot, so I kind of know. But I mean, upon your first time doing it, you definitely want to, use that reticle to your advantage. This is not Sunichia. This long guy in here, believe it or not. I think that's Leptocylindrus. That's another common species. But you can see how it might be confused for Sunichia just based on its how it looks. But we'll go through some more images next and show you why it's not. But I think this is a great picture to kind of show the different, on the same scale, all these the different sizes you're gonna see. And believe it or not, this is not even, I think there's even, like, they come even smaller than this. And this is one where the focus in the light, adjusting the light and the focus, it can really help because for me personally, if I have it a lot darker, like I have the illumination a little bit darker, I find that I can see the specks of kind of chloroplasts that are in the Sunichia a little bit easier. It doesn't make sense as I'm explaining it, but when you start to identify, you'll see that there are certain, lightings and focusing that can help you with your identification skills. So like I like to have it a little bit darker. Sometimes I'm looking at Sunichia so I can kind of see those spots more. Because I need to count each one of these. So, because there's maybe 8 in this chain here. And so usually when I'm counting, I'll count, like I said, there's usually 2 chloroplasts and then kind of a clear center. So I'll count like that section of chloroplasts and there's like a big break. Then I'll count the next section of chloroplasts. I'll show you another example. Okay, we're almost done, I promise. So those are the 3, main taxa that you're going to see, not necessarily most abundantly, but in terms of releasing toxins, those are the ones that you should be primarily concerned with. Something that you will see often, or pretty often, is Crinea mikimotoi, which is kind of this clover leaf shaped species here. It is related to the sister species of Crinea brevis, which is what causes the red tide in Florida. And Crinea brevis looks pretty similar, but it's a lot wider and a little bit more compressed laterally. So it's like kind of fatter than Crinea mikimotoi. So yeah, these kind of look like clover leaves. So they have these kind of distinct lobes here on the bottom. It has no thecal plates like Alexandrium does, so it's naked, so to speak. That's what they call it. Kind of has these yellow-brown chloroplasts in it, and like I said, the toxicity of it's not really understood in its effects on humans, so it's really more of an ecological concern. The other species that I haven't seen at all yet is Cocodinium polycricoides. It's also known as Margilophodinium polycricoides. It has 2. You'll find that with phytoplankton, there's usually multiple names. I don't really know how it works, but there's multiple names associated with, different species. But you may see it as marge, I don't know, it's Marge polycricoides, but I call it Coccididium polycricoides. And this is known for producing like brown tides or like rust colored tides. They have these yellowish green and brown chloroplasts here. They're kind of rod shaped and they usually form chains. Again, I haven't seen, I don't think I've seen one myself, myself yet since I worked here. That's why I say they're rarely seen, and I haven't heard of other, like, I get reports from Maine and Massachusetts and they haven't seen any either, or at least rarely. And they have this like distinctive, you can see it really clearly in this picture, this spiral shaped cingulum. So like it's not only displaced, but it's very displaced and it kind of comes around in this spiral here. So that's kind of distinctive of it. And you can kind of see it here too in this scanning electron microscope image, starting here, then wrapping all the way around. So you may or may not ever see that. And then lastly, we also monitor for Prorocentrum lima. You will see a lot of Prorocentrum micans, which is a like sister species to Prorocentrum lima. Prorocentrum micans does not release toxin, whereas Prorocentrum lima does release toxin. Again, I saw something similar to this last week, and that was the email I sent out that I was telling you about to my colleague saying, is this Prorocentrum lima? I'm not sure. And they confirmed, or they had their opinions that it was not. So it was helpful to have that kind of community, like I said. But this is another species we monitor for. It kind of has those, this image here helpful. You can kind of see all the pores. It has this central pyrenoid here, which is this circle, this large circle. It has kind of this depression, this V-shaped depression here at the top, interiorly. And it's just oval-shaped. Two species that hopefully we will never see here, but we do monitor for it because it's just required for us to monitor them by our like the guidelines that we follow for our program, our FTA guidelines. We monitor for Alexandriaum species, which again haven't seen it, and it's very distinguished by this. The cingulum is very wide. I think it's probably the biggest distinguishing factor, and it kind of has this. Kind of looks like a capsule, and they're very small. You can see this is 2 micrometers. And then, like I said, Carinia brevis, the species very common in Gulf of Mexico. Again, we haven't seen it here. Knock on all the wood that we never do. But yeah, it's very similar to the Carinia motoi, except that, like I said, it's a lot. It's like compressed and a little bit wider, but it does have these kind of yellow, brown chloroplasts. And then you can see in this image, I like this image a lot. This is from a lab in Florida. You can see the little nucleus in each of the lobes here. This is a typical view through a microscope. These could be Alexandrium. I just noticed that recently. But sometimes it looks like that. And this is an instance of when you would want to look at a diluted sample, or in this case, I probably would just look at like a 2-liter concentrated to 15 ml sample as opposed to anything being diluted. But again, like I'd give you the option. I'd give you multiple samples to look at, or I'd give you just a diluted sample to look at. So.
I'm thinking we're just going to be giving you samples that are countable and high.
Yeah. But like this is not even a bloom species, or not a bloom. Sorry, this is not a HAB, like a harmful algae species. This is Leptocylindrus, I believe, which is doesn't release toxin. But I would still, want to dilute the sample further. So this is it's a cool image, and this has happened frequently, and this has happened with like Sunichia, so that's fun. So. And then just my thanks to Eric again these were all his images. So all of the ones that I showed for the species, our specific species, were his. The ones that I obviously got from online, from him. And then these are all my resources that I tried desperately to fit onto one slide. All of Maine has lots of guides you can use, including the Alexandriaum guide. Some of the resources that we have is our SOP. I have a little density calculator where that table that I showed you, you can just plug in the numbers. Like you could plug in, I saw 5 cells, it'll spit out, and you can set up. I did a 1-to-3 dilution. I only counted 100 grids, and I counted 6 cells, and it'll spit out the cells per liter for you, so you don't have to do any math. Our data sheet that we use, which I don't know if you'll ever need, but it's just for you to see if you want. And then just some other useful guides that I think this one I don't have the link to, but the other do. So yeah.